Welcome to the MDS podcast, the official channel of the International Parkinson and Movement Disorder Society. I am Sara Camargos, Associate Editor of the podcast. Today, we are joined by Dr. Guillaume Cogan and Professor Alexis Brice from the Paris Brain Institute. They are the corresponding authors of the article selected as the Movement Disorders Review of the Year. The title of their article is Exploration of Neurodegenerative Diseases Using Long-Read Sequencing and Optical Genome Mapping Technologies. Congratulations to both of you for this important nomination. Dr. Guillaume, tell us a little bit about your background and what inspired you to write this paper.
Hi, thanks for having us. I'm a medical geneticist, and now I'm doing a PhD at the Paris Brain Institute. And we had a project together from the Paris Brain Institute with the National Institutes of Health, with our colleagues that are also co-authors of the paper, Kensuke Daida, Cornelis Bernhardt, and Kimberly Billingsley. So basically, we had a cohort of unsolved PD cases which had whole exome sequencing, and we had familial cases and also early onset cases. And we wanted to try to identify something like the mutation that is causing the disease. And to do so, we used long-read sequencing. I will go into the details of that in the future. And we had an interesting case. We wanted to report this to movement disorders, so we did. The editor asked us to do a review about long-read sequencing, but also optical genome mapping to get a review of the new technologies, new genetic technologies that we can use in neurodegenerative disorders.
Very nice. I think genetics can sometimes feel like a challenging topic for neurologists. So let's take a step back and start with the basics. We are going to talk about the type of variants that cause neurodegenerative diseases: single nucleotide variants, structural variants, and repeat expansions. Could you please explain these techniques used to study this kind of variants?
Yes, I can start and Guillaume will complete. I think what's really important is the fact that long-read sequencing means that you can analyze long fragments of DNA rather than having something like 150 to 300 base pairs read for each fragment. You have something like tens of kilobases. And this changes a lot because you can detect a lot of the rearrangements which are not detected by usual techniques. And this means, for instance, when there is an inversion in the gene, you can see the junction points, you can sequence them. You can detect more easily duplications or deletions, and that's a topic also for many neurodegenerative diseases. And as well, repeat expansions. The small ones can be captured by classical techniques, but once they exceed the size of 300 base pair, they cannot. So with long-read sequencing, again, you can pick up this type of variants. So that's the most important aspect, but there are other ones. You can, for instance, distinguish individuals who have two variants in a gene. You can say whether they are in cis or in trans. And for a recessive disease, they have to be in trans if you want to make sure that they are responsible for the disease. You can also have applications for gene and pseudogenes. When they are highly homologous, long-read sequencing allows, again, to sequence independently the gene. So that's really the basic. And we can certainly provide a few examples if you want.
Very nice. Especially when I read your paper, I found very interesting that you use these examples to show the advantages of long-read sequencing, such as you have mentioned pseudogenes and phasing. Could you please share with us the story about the siblings and twins with Parkin variants?
Yes, so our colleague from NIH, Kensuke Daida, first had these two siblings with a phenotype that was compatible with Parkinson's disease. So the siblings had an early onset, a slow progression of the disease. And using, I think it was exome sequencing, so short read sequencing, they had a pathogenic single nucleotide variant, but just one variant. So it's not sufficient to explain the disease. And then they used long read sequencing, and they identified an inversion in the second allele, which is explaining the disease. And the inversion was very large. It's a 7 megabases. So it explains why they didn't get it first. And then from our cohort, we had also two siblings with autosomal recessive form of the disease, compatible with Parkin, again with the phenotype. And we first used the conventional sequencing methods, multiple ligation probe amplification, targeted sequencing, whole exome sequencing. And we had a deletion of exon 4, but only one mutation again. So that was not sufficient to explain the disease. We used, again, long read sequencing, and we found that something very interesting. So on the first allele, we had a deletion of exon 3 and exon 4. And on the second allele, we had a duplication of exon 3. So overall, we have 2 copies of exon 3, which is normal, right? I hope we all have 2 copies of exon 3 here. I hope. And this is why the other sequencing tools were not able to see that. And only, let's say, only long read sequencing could identify this. And then Kensuke Daida and colleagues extended this study to, I think it was 23 individuals with one Parkin variant and an early onset of PD. And they were able to solve a quarter of cases using long read sequencing. So this is something that is meaningful, I think, and neurologists could think about it when they have a patient with one mutation in Parkin and a phenotype that is compatible.
And you were only guided for the phenotyping. This is very interesting. So you dig a little bit just to see if there was a structural variant that could explain the other in-trans variation.
Yes.
Is that correct?
Yeah.
Amazing. Besides discovering the new genes, another interesting aspect to explore is the repeat expansion characterization. How is this characterization relevant for understanding the phenotype or genetic counseling?
I think there are at least 2 aspects for the repeat expansions. First, it's the size, and appropriate sizing is very important because there is usually a threshold above which a repeat can be said to be pathogenic, and this varies a lot according to the disorders. And for some of them, I have to say that the threshold might be variable, is still disputed, but at least above a certain value, you're sure that it's pathogenic. So the first is the sizing, which is absolutely crucial for diagnosis. And the second aspect is the sequence of the repeat, because it turns out that at some loci, it's not only the size, but also the composition of the repeat, which is important. And there are alternative composition, some of which are pathogenic and others which are well tolerated and not associated with the disease. With long read sequencing, you get both with a single stone. You have both the size of the repeat and the composition, and therefore you can say whether the repeat is pathogenic or not. And this is very important for many of the recently identified repeat as FGF14 or RFC1, for instance.
Yes, maybe I can give examples about it. FGF14 is a very good example. So it's responsible for spinocerebellar ataxia number 27B. It was identified quite recently. And we know that expansions of GAA that are lower than 200, they are not pathogenic. However, when the repeat is above 300, we know that it's pathogenic. And using short read sequencing, because the size of the fragment of the read is about 150, we cannot know anything that is longer. We can just say that there is an expansion above 150, but we can't say if it's above 300 or not. So we can't say if it's pathogenic, and we need another technology to say that. But using long read sequencing, we can tell more precisely the size of the expansion, and we can say if the expansion is above the threshold or not. Another example of the importance of the motif, so it's a gene responsible for the well-known syndrome, cerebellar ataxia, neuropathy, vestibular areflexia syndrome, so the canvas. And you know that long expansions of AAA G, so it's 5 nucleotides, they are not pathogenic. However, expressions of motif AAGGG are pathogenic. And using long-read sequencing, we have the motif so we can know if it's pathogenic or not. And I think something also important is the presence or absence of interruptions. And we discussed this in the paper. So I think a good example is spinocerebellar ataxia 2. So it's a CAG coding expansion. And if in this expansion of CAG we have an interruption or several interruptions of CAA instead of CAG, we know it's responsible not for spinocerebellar ataxia, but Parkinson's disease. The motif is the presence or absence of interruption is important for the phenotype, but also for the age at onset, the penetrance, the inheritance of the disease, and the severity and the type of phenotype.
So we certainly can expect to identify more of these repeats in the future. And we know that in neurological disorders, they are particularly frequent. So I think that by sequencing many more cases, we certainly will discover unknown mutations in the future.
And also, you are able to check the methylation. You can understand a little bit more about the gene expression.
Absolutely. Using the main methods of long-read sequencing, we can identify the methylation. And this has, of course, several implications in diseases because usually, it's not always the case, but usually hypermethylated elements in the DNA have less expression compared to hypermethylation. And in the context of neurodegenerative disorders, I think this is interesting. For example, we have the gene. They studied this for NUTCH2-NLC, which is responsible for when you have a GGC expansion in the 5' region. It's responsible for a disease called neuronal intranuclear inclusion disease. And they found that it's very interesting. Unaffected parents of children affected by this disease had longer expansions compared to offspring. So it's not expected, right? But using long-read sequencing still, they found that this GGC expansion in the parents was hypermethylated. So the gene was less expressed compared to the lower expansion. And this expansion is pathogenic because it's leading to RNA foci that leads to a sequestration of RNA binding proteins. So basically, if you have a lower expression, you have less RNA foci, and then you don't have the disease. It's very interesting to see that methylation allows us to understand why some mutations are not pathogenic compared to others.
Amazing. Very interesting. Let's talk a little bit about targeting in long-read sequencing. In April 2024, we interviewed Professor Holding and Dr. Zombo Chen using targeted long-read sequencing to describe SCA4. Could you remind us how this method works and if there is another methodology for targeting instead of sequence everything?
Yeah, so it's a good question because you can use the long-read sequencing in several aspects. So you can do whole genome sequencing, but you can also do targeted sequencing. And I would say there are, in the paper, we talk about 3 methods to do this. So first, you can use a Cas9-based method. So you just cut the region of interest using CRISPR-Cas9, and then you only sequence this region of interest. So this is the first one. Another one is also simple. It's long-range PCR. So you amplify your region of interest using long-range PCR together. But of course, if you use this, you can have the amplification bias because you use a polymerase chain reaction, right? So this is the second one. And the last one is provided only by Oxford Nanopore Technologies. So the last one is adaptive sampling, and it's quite interesting. So basically, you have your strand of DNA, which is going through the pore, and the pore will analyze the first 100 base pairs. So for example, 400 base pairs, and it will see if these base pairs are in the region of interest that you told the sequencing machine to sequence. So if it sees that this fragment is not a fragment of interest, it ejects the fragment. So it will only sequence all the way through the pore fragments of interest. So those are the 3 methods: Cas9 based, long-range PCR, and ONT adaptive sampling.
Very nice. Professor Brice, I get the sense that long read sequencing is almost like the genetic version of deep phenotyping, kind of a deep genotyping. Do you agree?
I fully agree. With the deep phenotyping, you find things that you didn't see because you didn't look for them properly. And I think that's really important for neurologists to be able to perform this deep phenotyping, which can help diagnosing. And here it's exactly the same because the tools we used until long read sequencing were not able to detect some of these variants. And now that we have this tool, we can pick up these variants and we can improve diagnosis. So it's really exactly, it's very similar, except that the cost of deep phenotyping might be less than long read sequencing at the moment, at least.
Yes, speaking about the challenges, when it comes to long read sequencing, what are the big challenges for us besides the cost?
Beyond the costs, the challenges are first the wet lab. So the wet lab protocol is not yet standardized. Also, the bioinformatic pipeline to pull the variants is not standardized. And also you need a lot of data storage capacities because it generates hundreds of gigabases. So you need good storage capacities, also good GPUs and CPUs to call the variants. It's very computational intensive. And at the end of the chain, let's say, to interpret the variants, it's also harder because we have way more variants. At least we are confident in our variants because, for example, if you do short read sequencing, you try to analyze structural variants, you know that a lot of them are just false positives. But using long read sequencing, you know that most of them are true positives. However, when we analyze variants, if you want to identify the cause of the disease of a patient, you want to remove variants that have a high frequency, that are common, right, in all of us,
Yes.
only to select rare variants. The thing is that for long read sequencing, we don't have catalogs such as GenomeAD for short read sequencing. So it's hard to filter short read variants. Nevertheless, there are some collaborative projects, for example, the 1000 Genome Project, that is sequencing hundreds, if not thousands, of healthy controls to provide us population databases, allowing us to filter the variants according to the frequency. And finally, having a bit of experience with it, it's sometimes frustrating because you're like, I sequence all the types of variants. I have structural variants, SNVs, short order repeats, but I don't find the genetic mutation. But I know it's here somewhere. But I can take maybe one example. For example, we have intronic structural variants that may affect the splicing of a gene, but we don't have any tool to predict if it's affecting the splicing or not. So we hope that in the future, bioinformaticians will develop those tools so that we can finally identify the cause of the disease of all people with a genetic disorder, let's say.
Or even sequence all the RNA too.
Yeah, this is another. Using long read sequencing, yeah, we can identify new isoforms as we talk about in the paper.
And also in your paper, you explore the possibilities of optical genome mapping. Explain us how this technique works and what are the main advantages.
So it's quite different compared to long read sequencing because it's not a sequencing. So basically, you just label your DNA at some canonical sequence, short sequences, and then you have a microscope that is looking at the distance between your labeled tag of the DNA. So with that, you can identify structural variants that are above 500 base pairs. So you can't see anything below that. That's one thing. You can't also see SNVs, single nucleotide variants, using optical genome mapping. However, it's quite interesting because the depth, the coverage is better compared to long read sequencing. You can get 150 coverage depth using OGM compared to usually 20 to 30x with long read sequencing. And yeah, that's the main thing of OGM, I would say. So some people compared OGM and long read sequencing. And for long read sequencing, it's hard to identify variants, structural variants that are above 50 kb. However, OGM is good to identify those kind of mutations. So I would say if you have money, you can use both, and that would be the best. But you need money to do.
Of course.
No, I mean, you also need high molecular weight DNA, and that's
Yeah.
sometimes a limitation for these techniques. And classical biobanking sometimes use extraction techniques that do not provide such DNA. So it's something you have to take into account, at least for the prospective cohorts or samples that you are getting.
Yes, and something also for OGM, I think a limitation of OGM that is good to know for listeners is that it's very hard to get the precise location of breakpoints using OGM. You can't get it. Usually it's between 6 kb to 15 kb. So it can be important in medical genetics because using OGM, you can say that a structural variant is encompassing an exon, whereas it's not. So it's not a false positive, but it's just not as accurate for structural variants as long-read sequencing. So again, it's good to use both if you want to use OGM, yes.
So you predict these two technologies would or will become the first-tier approach to diagnose hereditary neurodegenerative disorders in the future?
Provided that the money is there to pay for them, yeah, I think that they clearly allow to solve a greater proportion of cases, and they are therefore very useful, I think, if we want to have an approach, a first and last tier approach, where a single technology can provide the results.
Yeah, there's still a long way to go to get these technologies as first tier, I would say. Yeah.
Yeah, very nice. Before we finish, is there one key message you both would like our listeners to take away from this paper?
Yeah, so I think you need to know your project and really what you want to see before using these technologies. And I think in our article, we tried our best to give examples so that the reader can understand what he can get from these technologies. And I think, yeah, reading this article is a way of better understanding this and use it in an appropriate way for someone's project. Yeah.
Yes, so doctors, thank you so much for joining us and sharing your insights today. Thank you to all our listeners for being with us on the MDS podcast. Stay tuned for our next episode, and until then, take care and goodbye.
Thanks for having us.
The views and opinions expressed by the participants in this podcast do not necessarily reflect those of the International Parkinson and Movement Disorder Society or their affiliated journals, Movement Disorders and Movement Disorders Clinical Practice. Any disclosures of the participants can be found within the episode description located on the MDS website.